Hey guys, it's Miss Adelini. I'm sorry I couldn't be there in class today. I am going to try this using an online video lecture, and I hope that it works. We are going to be learning about the macromolecules today using a Prezi that was created by Mr. David Nufke, so all credit goes to him. And I am going to be going through the actual Prezi. However, you have a notes document that goes along exactly with the Prezi. Those notes are the Unit 2 notes, Part 4, the macromolecules. In class together, we went through Part A, where we discussed the properties of the carbon atom and discuss why carbon is a really good atom for creating large molecules like carbohydrates, lipids, proteins, and nucleic acids. We looked at several functional groups like hydroxyl groups, carboxyl groups, carbonyl groups, sulfhydryl groups that are molecules that can be added to larger molecules to give them particular properties. For example, if a hydroxyl group is added to a large molecule, that large molecule is then called an alcohol. And typically, it is an overall polar molecule, meaning it has regions of positive charge and regions of negative charge. If a carboxyl group is added to a particular molecule, that carboxyl group then acts as an acid within that molecule, and it can donate extra hydrogen ions to solution. So the different functional groups can give different properties to larger molecules. So that's what we discussed together last class. We are going to be starting on the back of the third page of your notes where it says Part B, the macromolecules. We're going to be addressing 3 big questions within our lecture today, and by the end of the lecture, you should know some basic answers or be able to elaborate on these 3 questions. So the first one asks, how are the molecules of biological systems constructed? What they mean is, what is the basic structure of our 4 main macromolecules? Carbohydrates, lipids or fats, nucleic acids like DNA, and proteins. The second question asks, why are particular groups of macromolecules needed in biological systems? And by that question, we mean what are the functions of the 4 macromolecules within living organisms? And some macromolecules will fill several different functions. The last question is a little more abstract. It asks, how do the interactions of biological molecules lead to the emergence of life functions? So they're asking you how the structure and the function of those molecules help support the entire living organism. Let's go to the next slide. Okay, so the first thing that we have to ask ourselves is, what is a macromolecule? A macromolecule is a very large molecule within our cells. Macro meaning big. Most of our macromolecules, our carbs, our fats, our proteins, and our nucleic acids are made of the same atoms. Different macromolecules will contain the 6 major elements found in living organisms, CHNOPs: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The different combinations and bonding between those atoms will give us a particular macromolecule that has a certain structure and thus a certain function. Depending on how these atoms are combined, we can have extremely, extremely complex molecules like DNA that's posted at the bottom right corner. Again, we have 4 main kinds of macromolecules: the carbohydrates, which are sugars, lipids, which is another term for fats, proteins, and nucleic acids, the genetic material of the cell, like DNA or RNA. Okay. For the most part, macromolecules are made of building blocks that are linked together in chains. We call the building blocks monomers, since mono means one, so that is the single simplest unit. Poly means many, so a polymer is going to be a chain of monomer units, and each macromolecule is a polymer. The conversion or movement between monomers to polymers and back to monomers is facilitated or assisted by adding or removing water molecules. The process of actually creating a polymer is called dehydration synthesis. If you'll take a look at the picture, they show two molecules that are being added together to create a larger molecule. Since these guys are smaller, we'll call them monomers, but this one right here, I'm not sure that that would be considered a true monomer since it has 3 units. I think they're just showing us adding this monomer to this growing chain. And the way that we do this, the way that we create polymers by adding on monomers, is we will remove an OH, a hydroxyl group, from one molecule and a hydrogen from another, and in that process, water, H2O, is being lost and the molecule is joined together. Dehydration synthesis, if you think about the name, it actually makes a lot of sense since dehydration is lacking water. So in this case, we are losing a water molecule, and synthesis means to make. We are synthesizing a larger polymer. All 4 of our macromolecules are put together using this process of dehydration synthesis. This type of reaction is considered anabolic. An anabolic reaction is one where we are building larger, more complex molecules from single building blocks. And this process requires energy. Without energy, we could not link those monomers together to make a polymer. It also requires the assistance of certain helper molecules or enzymes, and we'll discuss what enzymes are and how they work later on in the unit. That corresponds to objective 5 for our unit, and they're a specific type of protein molecule. We'll be addressing enzymes in our Unit 2, Part 5 notes. Hydrolysis is going to be the opposite process from dehydration synthesis. Instead of building a polymer, we're breaking a polymer down into monomers. Instead of losing a water molecule, we are adding a water molecule to facilitate the breaking of that polymer. If you break down the word hydrolysis, that makes sense since hydro means water and lysis means to break. So we're using water, hydro, to break or lyse that polymer molecule. Unlike dehydration synthesis, which is an anabolic reaction, this process, hydrolysis, is a catabolic reaction, which means it breaks down larger molecules and reduces complexity. A more complex molecule might be a polymer. A less complex molecule is a monomer. In this process, energy is released, so it doesn't require a whole lot of energy to get started. However, there are helper molecules that are required to assist with this process, and these are different types of enzymes than you would see assisting a dehydration synthesis reaction. Okay. We're going to be taking a look at each of the 4 macromolecules in turn. Things that you're going to need to know for each of the macromolecules is the overall structure of the macromolecule, its function in living organisms or multiple functions, the elements or atoms that are found within that macromolecule, the name and examples of the monomer or the building block for that macromolecule, and the name and examples of the polymer or long chain that makes up the macromolecule. We're going to start with carbohydrates, go to lipids, then we will go to proteins, and we will end our lecture with the nucleic acids like DNA and RNA. All right, guys, we're back. So again, the first one we're going to take a look at is the carbohydrates. Little general information for you about the carbohydrates. These consist of simple sugar molecules as well as more complex polymers like starches. There are three main elements that are found within carbohydrates: just carbon, hydrogen, and oxygen. Because oxygen is present and it's highly electronegative, it holds electrons to itself very tightly and therefore ends up with a slight negative charge, whereas anything that it's bonded to will typically have a slight positive charge. The overall sugar molecule is going to be polar, since it has regions of negative charge and regions of positive charge. Within sugar monomers, so the single units, we always have a certain ratio of carbon to hydrogen to oxygen atoms. We always find them in a carbon 1 to hydrogen 2 to oxygen 1 ratio. Carbohydrates are used for short-term energy storage, in contrast to lipid molecules, which are used for long-term energy. When your body doesn't have enough sugar molecules to break down, it will start breaking down your fats. But here we're just looking at sugar molecules for short-term energy. And we also see some of them used for structural purposes. There is a large carbohydrate called cellulose that's found in the cell walls of plant cells. And that's used to help maintain plant cell structure in the cell wall, partially so that the plant won't wilt. It'll be able to grow up towards the sun in order to get the sunlight it needs for photosynthesis. Another large carbohydrate that's used for structure is chitin. Chitin is both found in the cell walls of fungi as well as the exoskeleton or outer crunchy skeleton of insects like cockroaches. So both cellulose and chitin are two large carbohydrates that can be used for structural purposes. We call the monomers of carbohydrates monosaccharides. Saccharide can be kind of a difficult term to remember. I always think of saccharin, which is the artificial sweetener that's found in diet sodas like Diet Coke, Diet Dr. Pepper. It's a problem, I'm addicted, but whatever. And because saccharin is an artificial sweetener, that helps me remember that the term saccharide means sugar. So monosaccharide, when you break it down like that, really does make sense. Mono means one, saccharide means sugar, a single sugar molecule. We can have sugar monomers that have slightly different structures and sizes. The smallest sugar monomer that I know of has 3 carbons. You have 2 3-carbon sugars pictured at the bottom here, glyceraldehyde and dihydroxyacetone. I think I pronounced that right. And because they have 3 carbons, we call them triose sugars, tri meaning 3. And they distinguished in this image between a sugar that has a carbonyl group, the C double bonded to the O, at the end of the molecule. So that would be an aldehyde sugar, since aldehyde means the carbonyl group is found at the end, versus dihydroxyacetone is an example of a ketone sugar, or a ketose. That's going to be one where the carbonyl group, the C double bonded to the O, is found in the middle of the molecule, not at either end. Usually, sugars are going to have the last three letters as OSE, but these two right here, for some reason, do not. They just want to make us unhappy. You have an image of those two sugars. It's slightly different in your notes, but it still works. On this PowerPoint that I'm showing you right now are two pentose sugars, pent meaning 5. So these guys both have 5 carbons, and they're showing you an aldehyde pentose version where the carbonyl group is at the end, and they also show you a ketone pentose version where the carbonyl group is in the middle of the molecule and not at either end. On your pictures, you have one 5-carbon sugar present, and that's fructose, or fruit sugar. They show it stretched out with the carbons in a line, but in reality, in nature, that molecule forms a pentagon with the different points of the pentagon being carbon molecules or carbon atoms. So in nature, that 5-carbon sugar is going to form a ring. Ribose, one of the ones that they picture here, is the sugar that's found in one of our nucleic acids, ribonucleic acid, or RNA. We also have another pentose sugar, deoxyribose, that forms part of the backbone of the DNA molecule, deoxyribonucleic acid. The ones that you probably think of most, oh my goodness, I made a mistake. Oh, Adelini, I'm so sorry, guys. You can make fun of me all you want. Fructose is actually a 6-carbon sugar. So you may want to make a note of that in your notes. It does show it as a pentagon because some of the carbons are actually sticking off of the molecule. So I apologize. Fructose is a 6-carbon sugar, and that's our fruit sugar. So again, change that in your notes so it says fructose is another 6-carbon sugar, or hexose. Hex means 6, so our hexoses would be 6-carbon sugars. And in addition to fructose, we also have glucose and galactose pictured here, and both of those will also form rings in nature. Okay. Again, the ones that you probably think of when you think of a sugar monomer or monosaccharide are the hexose sugars. You've probably heard of glucose and fructose before. Probably haven't heard of galactose, but galactose, when added to glucose, gives us a very famous sugar, lactose, which is found in milk. And some people have lactose intolerance, which means they have the inability to digest lactose, which is found in cow's milk, and they won't tolerate cow's milk very well. In order to build larger sugar molecules, we can combine these sugar molecules, these monosaccharides, using dehydration synthesis. We will add those molecules together, and in the process, we will lose a water molecule. If you put 2 monosaccharides together, you get a disaccharide molecule, di meaning 2. And there are 3 that are listed here. If you add 2 glucose molecules together, it gives you maltose or malt sugar. If you put glucose and fructose together, it gives you sucrose. You've probably heard of that one before. That's table sugar. That's what you put. You guys shouldn't drink coffee, but if you did drink coffee, you would put that type of sugar in your coffee. Glucose and galactose, when you put them together, make that milk sugar that some people have the inability to digest. And when we're adding these monomers together, we're always doing it by dehydration synthesis. Okay. If we're linking many sugar monomers together, we create what's called a polysaccharide. Poly means many, so this is the polymer for a carbohydrate. We have two polysaccharides that are used for energy storage, and those polysaccharides are amylose or starch, which is found as the energy storage molecule in plants, and glycogen, which is found as the energy storage molecule in animals. Glycogen in animals, or in humans specifically, is stored inside our livers, and when we're exercising, our body will release that glycogen from our livers, break it down into glucose, and then use cellular respiration to convert glucose to ATP, a really quick source of energy. The other polysaccharide that we mentioned before is cellulose. It's found in plant cell walls, and it has a very strong structure that enables it to assist with the hardening of the stem. Now, starch and cellulose are both made out of chains of glucose monomers. However, these molecules are not the same, and they do not have the same function. One is used for plant energy storage, the other is used for plant structural support. So we have to look at the differences between these two molecules and how they give them different properties. I drew you a picture of a cellulose molecule and a starch molecule and showed you how the glucose monomers are linked differently in starch versus cellulose. These are the annotations that are located on your notes, and you can also see good pictures if you go to page 73 in your textbook. This is like the one and only time I'm going to tell you to use your textbook. Let's see if they will give us, nope, they won't. I thought they might give us a nice picture. I'll try to zero in on it a little bit, but I don't think it will let me. Let's see. Oh, yeah. Oh, Adelina, you're so smart. Here we go. Okay, so up top, they show us a starch molecule. We can tell that those are glucose monomers linked together because they are rings, they're hexagons. Starch contains alpha glucose monomers. Alpha glucose just means that the glucoses are linked together so that the direction of the bond between the glucoses is the same each time. Notice that my bond is pointing down, the oxygen in between it, between my first two monomers. It's pointed down with the oxygen in between it for my second two, and it's pointed down with the oxygen in between it for my last two. We call these 1,4 linkages because we are linking the first carbon in one glucose monomer with the fourth carbon in another glucose monomer. The bonds that link the glucose molecules together are a special type of covalent bond found in carbohydrates only called a glycosidic bond or a glycosidic linkage. Again, this bond is formed through dehydration synthesis. Now, cellulose is a little bit different. We say that the glucose monomers here are beta glucose monomers. All that means is when the glycosidic linkages, or the bonds between our monomers form, they're in opposite orientations to their neighbors. So this first one points up, the second one points down, and the third one points up. We're still linking our one carbon on our first glucose monomer to the fourth carbon on our second glucose monomer. However, each linkage or each bond is pointing in a different way. As a result of how their monomers are connected, starch and cellulose are going to have different overall shapes. Starch, when it has a long chain of monomers, forms a helix or a spiral structure, whereas cellulose, as a result of those alternating or opposite glycosidic linkages, is able to form a long straight chain that can hydrogen bond with other cellulose molecules that lie parallel or next to it. This forms, when you have a bunch of those cellulose molecules, a very strong cable-like structure that's seen in the plant cell walls. That makes it a very strong building material and very useful for structure. Okay. Cellulose. All right. Now, certain organisms are better than others at digesting cellulose. It's a very tough material for your stomach to break down. And if you eat a lot of vegetables, it's probably not too good for your stomach. Herbivores eat a lot of grass, so they do need to be able to digest a lot of cellulose, and cellulose that hasn't been softened or broken down by actually heating it. So cows, which are called ruminants, have a special digestive tract. Their upper gastrointestinal tract is very large, and they have several chambers to their stomach. They're able to mechanically digest or actually crunch the cellulose as it goes through their stomachs. They're also able to regurgitate and re-chew their cud or their partially digested grass to further digest the cellulose within it. And they also have bacteria located in their intestine whose specific job is to help them break down that cellulose. Termites are another example of an organism that's pretty well adapted to breaking down cellulose, since all it does is eat wood, which has a lot of cellulose in it. They have a symbiotic relationship with a small single-celled organism, a protist. The termite gives the protist a real nice place to live in its gut, and the protist helps with the cellulose digestion. Now this is the grossest one, cecophores. Cecophores means poop eaters, and bunnies or rabbits have a very expanded lower GI tract, gastrointestinal tract, so their intestines do a lot of the work breaking down the cellulose. They can't regurgitate their food like a cow could. However, they can poop out the partially digested cellulose, eat it again, and continue to digest it on the second round through. It's disgusting. I don't like it, but they do it. Oh yeah, we're going to skip through this. So this, oh wait, we'll go back. This shows you what the cecal pellets, which have only been through the digestive tract once, might look like. And they will re-eat those, and they'll poop out real fecal matter after the second digestion. Nasty. The other carbohydrate that I mentioned was chitin. That's also a polysaccharide, and we mentioned that it's used in fungal cell walls, in the exoskeletons of arthropods like insects. And one thing I didn't mention is that chitin is actually used in stitches that are supposed to dissolve. I don't know if you've ever had dissolving stitches before, but they do not dissolve, but they're supposed to. The last type of polysaccharide that I'll mention is peptidoglycan. This is found in bacterial cell walls and is used for structure. I'm going to take a break. I'll be right back. Okay, so now we're going to talk about the lipids, another term for fat molecules. and we'll start with some general information about the lipids. Lipids include fats, oils, waxes, and they're made out of three main elements, the same elements that are found in sugars. The difference between carbohydrates and sugars, however, is that carbohydrates and lipids is that lipids have mostly carbon and hydrogen. They only have a couple oxygen atoms. Carbohydrates have more oxygen atoms in comparison to our lipid molecules. Because lipids are mostly carbon and hydrogen and have very little oxygen, overall they are non-polar or hydrophobic molecules. They're used for a couple different purposes within organisms. Two main purposes are for long-term energy storage as well as insulation, keeping the organism warm. Here it says that they don't have any polymers. I'm not sure I really like that. I think what he's trying to say here is that we don't have actual chains of single building blocks making a large polymer. Instead, each large molecule is made up of just a couple pieces. We have three main groups: triglycerides, which are the fats that we know of; phospholipids, a lipid molecule found in the cell membrane; and also steroid molecules. Steroid molecules are found in our bodies. These are not anabolic steroids. They're not the steroids that you take to buff up. These are natural molecules within your body that are used for signaling purposes. A couple examples are testosterone and estrogen, the two sex hormones found in males and females. Okay. The basic structure of a triglyceride molecule is two parts. One glycerol molecule. I want to see if it'll zoom in for me. Here we go. Okay. So if you take a look at this image, it shows the glycerol molecule on the left-hand side surrounded in gray. And the glycerol is really going to be the only part of that triglyceride molecule that has oxygens in it. For that reason, the actual glycerol molecule is polar, but since it's so small in comparison to the entire molecule, it does not give the triglyceride polarity. The overall triglyceride is still nonpolar. And that's because of these three orange molecules, which are called fatty acid chains. Fatty acid chains just have a bunch of carbon atoms bonded together with hydrogens branching off of them. We call this a triglyceride because there are three fatty acids, and each of our fatty acids will join with the glycerol molecule through dehydration synthesis. So they'll link up to glycerol, and in the process, a water molecule is lost. The specific covalent bond that's formed during dehydration synthesis to create a lipid is called an ester linkage or an ester bond. Remember, when we're talking about carbohydrates, we call that covalent bond between sugar molecules a glycosidic linkage or glycosidic bond. But in this case, it's an ester bond since it's a fat. No, go back. All right, we have two types of lipid molecules depending on how the carbons are attached to one another. These two types of lipid molecules are called saturated versus unsaturated. It has to do with the bonding of carbon atoms to one another within our fatty acid chains. If it's a saturated fatty acid, that means that there are no double bonds between adjacent carbon atoms. And if we go back up to our picture, this particular triglyceride molecule, it appears that all of the fatty acid chains are saturated. there are no carbon-carbon double bonds. Within a saturated fat, the fatty acid chains are going to be very straight. Therefore, they stack neatly on top of each other at room temperature. And because it's so dense when they stack like that, they're going to be solid at room temperature. The bad thing about those saturated fats is that they stack and clump within your arteries and veins, so it could cause plaques to form in your arteries and veins and potentially a heart attack or stroke. Unsaturated fats have at least one double bond between the carbon atoms in their fatty acid chains. If you take a look at this image of a triglyceride molecule with a single unsaturated fatty acid chain, they show the double bond that's supposed to be located between two carbons. The location of that double bond produces a kink in the fatty acid chain or a bend. Because of that, unsaturated fats don't stack nicely on top of one another, and since they stack loosely, they're going to be liquid at room temperature like a vegetable oil or a fish oil. Those are going to be much healthier for you because they're not going to clog your arteries. And in fact, you do need some fats in your diet. You should try to make sure that those are unsaturated fats. Phospholipid molecules are our next type of lipid. They are similar to the triglycerides, but they are not exactly the same. Instead of having 3 fatty acids, they only have 2 fatty acids. They also have a glycerol molecule just like the triglyceride, but they have an additional phosphate group on top. Different regions of this molecule are polar versus nonpolar. The head that includes the phosphate and the glycerol is polar, it loves water, but the fatty acid tails themselves are nonpolar. They do not like water. Within a cell membrane, we see a lot of phospholipid molecules. Here they're shown with their polar head region as an orange circle and their nonpolar tail regions as 2 yellow chains or tails. Notice that they arrange themselves as a double layer that surrounds the cell. That's because there's water outside the cell, which the heads want to be close to, and water inside the cell, which the heads on the bottom layer want to be close to. The tails want to get away from water, so they isolate themselves at the midline of that membrane. Okay, steroids are our last group of lipid molecules. The basic structure of a steroid is a combination of 4 carbon rings made out of a bunch of carbon atoms arranged in a ring. So we've got 4 rings, and then we'll have different functional groups like hydroxyl groups, carbonyl groups, hanging off of that basic 4-ring molecule. The different functional groups give us the different types of steroids, and in fact, 2 of our steroids, testosterone and estrogen, differ only in a couple functional groups that hang off of that 4-ring molecule.